Thanks so much. I'm really excited to be here to tell you about my research. I'm originally from Kansas City. I did my undergraduate work in chemical engineering at Florida A&M University, which is a historically black college in Tallahassee, Florida. I then moved to Chicago or Evanston and did my graduate work also in chemical engineering at Northwestern University, moved on to a postdoc at Johns Hopkins in biomedical engineering, and now I'm at USC. I've been there for just under 3 years, and I'm really excited to tell you about the work that we're doing. I run this lab for computational systems biology. We have 3 really overarching goals, broad goals, which are to understand and examine the dynamics and regulation of biological processes. We want to be able to synthesize and interpret clinical and experimental data that's available with the ultimate goal of supporting the development of novel therapeutic agents. So we have 3 primary projects in the lab. One has to do with studying angiogenesis signaling in breast cancer tissue. And in this model, we have pro- and anti-angiogenic factors that we're looking at the signaling of. We also have another project that looks at metabolic reprogramming in pancreatic cancer cells due to the large stromal compartment in the tumor. And then lastly, we're looking at T-cell signaling for cancer immunotherapy. So I have just a little bit of time to tell you about a small part of one of these projects. And so I'm going to talk to you about the systems biology model that we're putting together to help understand and better predict the signaling dynamics that occur in CAR-engineered T-cells. This is in collaboration with a couple of other researchers at USC in the Department of Chemical Engineering. And so I probably don't have to tell you very much about this system, but chimeric antigen receptors, or CARs, are engineered receptors that are now can be expressed on T cells. They recognize an antigen that the tumor cells are displaying on their surface. They're comprised of this single-chain variable fragment that's derived from an antibody that recognizes a specific antigen, and then they're also combined with different intracellular signaling domains. Now, these therapies have been very effective in B-cell lymphomas, even leading to complete remission in some patients. And that's really a dramatic and durable response, but there still are some limitations to this cell-based therapy. And some of these are listed here, where they can induce cytokine release syndrome due to overactivation of the immune system. There can be on-target but off-tumor effects when trying to adapt these therapies to solid tumors. Also, low antigen concentrations are such that they perhaps can't stimulate the CARs themselves. So this is a very active and exciting area of research, which is developing next-generation CARs, those that are targeting multiple tumor antigens, as well as those that are incorporating chimeric inhibitory receptors. So a lot of that work has really been done in sort of this guess and check, trial and error approach. And so we're trying to put a little bit more mechanistic understanding about what's happening. How can we distinguish between the signaling that's being activated by different CAR constructs? And to do so, we're applying this systems biology approach, which again can help us optimize and understand CAR T-cell activation, and then perhaps develop a set of design parameters for the optimal design of these cell-based therapies. And so what we're looking at are CARs that have the CD3 zeta chain with or without different co-stimulatory domains, including CD28 and 41BB. This is a schematic of the model network that we've put together. It's very complex, a lot of interacting parts, a lot of positive and negative feedback mechanisms. But what we have is this CAR we're simulating that can then lead to activation of the MAPK signaling pathway, which leads to proliferation. We also have AKT for pro-survival pathways. And so this is really exciting what we're doing. Of course, if you have any type of model, then that means that you need quantitative data to train the model, to validate it, and to test the model predictions. And so these are ongoing experiments in my lab in collaboration with others at USC, where we are stimulating JERCAT cells that are engineered with the CARs with K562 cells that are expressing the CD19 on their surface. We can look at different CAR expression levels, different CD19 expression levels, and then we can also look at what's happening in a more controlled setting in the in vitro reconstituted liposome membrane system that was recently developed by Hui and Vale. So these experiments are ongoing, but I do want to tell you just a flavor of the type of computational modeling that we can do. So these are data that are taken from a very historic paper, 2005, from Alton, Bennett, and Germain, which measured the time that it takes for 50% of the cell, 50% of ERK inside of cells to become doubly phosphorylated as a function of the antigen concentration that the T cells are presented with. And this is just endogenous T cell receptor signaling data. So you can see that there seems to be an optimal antigen concentration above which or below which there is a longer response time. And so what we wanted to do is just make sure that our model is giving us results that make sense. And so we have fit the model just a subset of the parameters. We're just looking at 6 parameters from endogenous T cell receptor signaling. We selected those parameters because we did a sensitivity analysis, which really helps us understand what are the most influential parameters in the model. We selected 6 of those influential parameters and fit the model to this data. And you can see we do a very nice job of even capturing that non-intuitive sort of optimal concentration for the antigen. But then we took it the next step in order to see, okay, what happens if we change out different intracellular signaling domains? And so the baseline model in endogenous T cell receptor signaling has the CD3 zeta chain with 3 immunotyrosine activation motifs, or ITAMs. We exchanged that in our model for one that just has a single ITAM. You can see that for low antigen concentrations, the T cells are not even becoming activated. And even this optimal concentration seems to be even more pronounced in this case. And then lastly, we looked at what happens when you have the CD3 zeta chain along with CD28 to see what happens when you add in this co-stimulatory domain. Very excitingly, the model predicted without any tuning of the parameters that the response time decreases. So that co-stimulatory domain really is speeding up the response time in terms of activation of the T cells. We were excited to see this. This is just initial model exploration, and it's exciting to show that the response time does truly depend on the structure of the signaling domains that are involved, and we make this analogous to the CAR structure as well. So of course, everything is still ongoing. We have the model present. We're collecting the data. It can distinguish between different CAR constructs, and we can use the model as a quantitative, robust tool in order to perform different in silico trials to investigate the effects of CAR expression, the effects of the antigen expression, and so on. So with that, I'd like to thank the members of my lab, graduate students and postdocs, a very talented group of undergraduate students that I've had the opportunity to work with, and of course thank all of you for your attention. I'm really excited about this work, and if you want to talk more about it, we can have discussions this afternoon. I'm happy to sort of invite you to USC or come out to your institutions as well. I think this is just an amazing opportunity to present this work to you.